Okay, so in order to ensure that you're going to get a grade 9 on your science, or a very good grade on your science, you need to ensure that you know all the required practicals, as you don't know which one is going to come on the paper. And normally there are two questions that are core practicals or required practicals. So in today's video, we're going to be looking at osmosis in potato strips. So what you have, or what you did in school, is that you have potato strips here, as shown here, and they're placed in different concentration of solutions, okay? And as you can see here, we are using a sucrose solution. However, it can be a salt solution, okay? And it doesn't have to be potato strips, but we're just investigating osmosis. Now, what happens is that you take the mass of the potatoes before and after. So you leave them for a while, the longer the better. And for after you've taken the mass before, you then look at the mass after and to compare. And then what you notice is that some of the potatoes may be bigger or some may be smaller. Now, before you take the mass readings, you have to ensure that you blot dry the potatoes. Very important that you mention that you blot dry the potatoes. That's a source of error. Because what you don't want happening is to squeeze any water that may be inside the potato out. So you're just removing the surface water. When doing this, ensure that you know the variables, okay? So your independent variable is the concentration of the solution, okay? So your independent variable is the concentration of the solutions, okay? Your dependent variable, and by the way, independent means what you're changing in the experiment. So your dependent variable is what you're measuring. So you're measuring the change in mass, okay? And your controlled variable is what you're keeping the same. So your controlled variable is what you're keeping the same. And what you're keeping the same is the volume of the solution, okay? You're also keeping the temperature the same, okay? All right, so ensure that you know about the variables. So to calculate the percentage change in mass, so at the end of the experiment, you need to work out the percentage change in mass so that you're able to compare the results. So you have your balance, which you use to take the mass before, you blot dry, then you take the mass at the end. Okay, and this is the formula for calculating the percentage change in mass. Okay, ensure when you're studying that you know the definition for osmosis. So osmosis is the movement of water from a high water concentration, dilute, to a low water concentration, and ensure that you're mentioning that it's going through a semi-permeable membrane. Now, after you've looked at this practical and you've made your notes, it's good practice to then have a go at a question. So let's have a look. A student investigated the effect of different concentration of sugar solution on pieces of potato. Here are all the steps. What is the independent variable? So remember, independent variable is what you are changing in the experiment, okay? And we are changing the concentration of the sugar solution. That's what we're changing, okay? Very easy, too easy. All right, so they've given you some data here, and they said explain, very important, so explain why the potato in 0.0 moles per dm cube sugar solution increased in mass. So if you have a look at the mass, it was 7.94, it's now 10.12. Why did it increase in mass? Now, increase in mass, remember what we're doing osmosis, so it increased in mass because it gained water. Okay, I would say that's a mark to mention that it gained water. Now, the question has asked you to explain, so we have to go one step further. What process did it gain water? It gained water by osmosis. Okay, and that's your next marking point. Okay, you can use bullet points in your exams, okay? So gain water by osmosis, right? And if you want to go one step further, you could then explain what osmosis is, but you don't have to for this question. All right. Next up, they've now asked you to plot a graph of the results. So you have your concentration here, you have your change in mass here. They've done 3 points for you. Okay, so we have to do the others. So they've done 0.3 for you. Okay, so we need to do 0.2. So we're here and we're doing change in mass. So that's 0.25. So each one is worth 0.1. So 0.25 is going to be here. 0.1, 0.25. Okay, or we can just use an X that they've been doing. All right, good. And then 0.1. So that's 1.15. So 1.1, 1.2. So 1.15 is going to be in between here. And then finally 0.0. That's 2.20. 2.1, 2.2. Okay, and then they now ask you to draw a line of best fit. Okay, so my line of best fit, a line of best fit doesn't have to be a straight line. My line of best fit can be a nice smooth curve. Okay, good. Then in this scenario, yeah, that's more looking like a curve to me. Okay, then the next question then says the mass of a piece of potato does not change when the concentration of solution inside the cells is equal to the concentration of solution outside the cells. That's important. That's what you call isotonic. That's another way they could ask that question. So determine the concentration of the sugar solution inside. So the mass does not change. So we're looking for where you have 0% change. And I'm thinking that's roughly about 0.24. Okay, thereabouts. Yeah, 0.24 moles per dm cube. All right, and finally, we spoke about this percentage change in mass. It's a 3 significant figure, 3 marks. That's an easy 3 marks. Okay, for the 0.2 moles per dm cube. So let's look at that. Do we have the final mass? Yes, we do. So the final mass is 8.21. The initial mass is 7.96. Okay, you're going to divide all of that by 7.96, which is the mass at the start. You're going to times that by 100. And if you've done that correctly, you should get on your calculator 3.1407. Remember, it says to 3 significant figures. So it's going to be 3.1. That's going to be 3.14. Okay. It's going to be 3.14. Okay, and that's the percentage change in mass. Okay, and that's your answer right there. So remember, to get under grade 9 in GCSE, you have to go through all the core practicals, do past paper questions, okay, and just remember to like my content, share my content with your friends, and go over to my YouTube channel. Okay, all the best in your GCSEs.